I'm Jim Sluka. I'm going to introduce the speakers for this last session. I believe it's our last session. I am involved in the IU version of the ontology development, and in the coming year, I'm going to take over largely for what Paul Macklin has been doing. Paul will still be around on these occasions. So our first speaker for this section is Katy Börner. I think everybody knows Katy because she's been talking all morning. Katy is a member, a professor of information science in the Department of Information and Library Sciences here at IU. She's also part of the core faculty in cognitive science and an adjunct professor in just about every other department on campus. She's widely known for her work in developing advanced visualizations across a wide range of knowledge domains. And Katy is, of course, the lead PI in IU's part of the HuBMAP project. Katy?
Thank you, Jim. So as you saw, we tried to have always a HuBMAP presentation in the beginning of each of those 4 sessions, just so that you get to see our world, our very young world after 7 months. And here you have the presentation on the multi-modal CCF user interface for data providers and for data users as part of what our team has accomplished so far. And also a little bit of an outlook of where you want to go next.
Is it not working?
So overview, I will talk a little bit about the data. Some of you might have seen Paul's presentation this morning. Then I will talk about 3D spatial models linked to terminology and ontology. Again, some of this was captured in Paul's talk. I will talk about 2 interfaces. One is for CCF tissue registration, and the other one is for data users for CCF access to data. I will also have a little bit on our year 2 plans and open questions. In this overview of the data pipeline, you see that we in HuBMAP have multiple TMCs. Right now we have 5, which generate organ-specific data, which then some of this data makes it to the Pittsburgh Supercomputing Center. And again, a subset of that data makes it to the CCF construction teams and the user interface that comes with the CCF. There are lots of other elements in this figure that I won't have time to go through, but what I will do is to show you a tissue registration user interface that's used by the tissue mapping centers ultimately, if we can get it to work. This is really an exploratory study right now. And on the right-hand side, we have a CCF user interface, which uses the CCF ontology and metadata that you learned about from Paul's talk, and also data provided by the Vanderbilt team to show how semantic search, filtering, and browsing might work. In terms of data, again, Jeff gave a really good overview this early morning on the data they have provided so far. The data comes also with clinical metadata. They have provided us with information on their favorite cell types and cell states, which is very interesting and relevant because HuBMAP ultimately aims to map at the single cell level. We also work closely with Shin Lin at UW. And in year 1, they will have tissue data for 1 to 2 centimeter cubed volumes from 9 different sites from a human heart from 1 individual. They also have shared their data dictionary and their cell types. And as you will see, there's quite a bit of difference in the data formats and also in cell types. If you, for instance, look at the clinical data, sometimes age is provided in years, sometimes in months. So all this needs to still be standardized across the HuBMAP consortium. And there are, again, many more organs involved. For the CCF, we have started to have the CCF data wiki, which ultimately will help us define a minimum information standard that's needed for the CCF construction and usage. If you are part of the HuBMAP team, you should be able to access this link. And again, you have to have a GitHub account, and then you might have to also contact Bruce Hare to actually then get added to that list. So this is not easy right now. I think we are trying to get a better solution. Ultimately, what we are hoping to do is to have a kidney here. This is actually the kidney from the NLM 3D data set. Which has ultimately also anatomical features and submolecular features. And then you just extracted a tissue sample, and we want to make it easy for tissue mapping centers to help us understand where that tissue slice came from. In HuBMAP, we have currently 10 organs, 13 different data types generated by 25 different technologies. It's interesting to note that many of these technologies are still under development, protocols are optimized, so this is not static at all. And it's actually kind of tricky to keep track of all of it. And so again, that's where the CCF data wiki comes into existence and into effect, because we can have every TMC take care of their own protocols and of their own ways of registering their tissues in their favorite organs documented there. And a registration that might happen very well for the kidney might not work at all for the lung and might not work at all for some other organs. So I think we have to be very organ specific with the registration. This is a slide which also Sanjay had in his slides. You see here the enormous diversity in just kidney structures. So this is a set of 492 kidneys, and as you see, some of those kidneys have a total number of 6, 7, 8, 9, or 10 renal calyces. And so the big question becomes, what should the reference system represent initially? Maybe it just represents 8 because that seems to be the highest bar. But ultimately, of course, we want to capture as much diversity as we can afford with the budgets we have. In terms of how the registration is currently done, typically the pictures are taken from the dissection process. There are also manual annotations of different areas. And here on the right-hand side, you see the 9 sites from which Shinlin is planning to extract tissue samples. We believe that a combination of rough placement of by human experts, here a surgeon, and then 3D pattern matching, plus fine adjustment using machine learning ultimately, but initially using also human intelligence so that we have training data, will give us the best results of registering tissue samples with the CCF. We will measure error from the precision of tissue sample procurement and placement in these 3D browsers. We also hypothesize, and that needs to be tested, that placement accuracy will considerably improve if we add additional information into our 3D models, such as landmarks, major scaffolds, MR, CT scan data, etc. And maybe even information on protein, lipid, and other structures, which we just saw beautifully in Seth's talk. On the right-hand side here, you have the NLM model of the human heart, of the visible human, the male version. So again, this heart model is actually reconstructed from that original visible human data set. Kristen, which works at NLM, did this 3D extraction and composition, and what you can now do is to rotate this in 3D and then place the samples that you have just surgically extracted in the right areas in a rough way, and then after the assays have been run over that tissue sample and you have more information on the internal structure of that tissue sample, you can also use that information to do a better fine placement. And again, after a while of doing that, maybe 50, 100 times manually, I think machine learning algorithms can help with that as well. So this is for the heart, and you see the red cubes here correspond to the extraction sites that Shinlin is planning to use. Here you see a kidney sample, and it's the same idea again. So you take a kidney sample, and this is again planned for year 2, but we have some first prototypes running, and it would have to be placed where it was extracted. This is our PhD student, Andreas Böckler. I'm very proud to actually have that all working so that you can actually take that and kind of try to align it. And right now, these are random dot patterns because we don't have the data yet. But as soon as data becomes available, I think this will look almost as gorgeous as what that just showed. So that's for the tissue registration part. But the main focus of today's presentation is the CCF user interface. here in our initial 7 months so far, but by the end of 9 months, we will have a fully functional initial prototype, which is meant as a conversation starter and to show what data is available so far within HuBMAP. This user interface is supporting a researcher who is interested to search for, filter out, and review and download biomolecular data in the context of the whole human body, or in spatial relationships to specific organs, tissues, or cell types. The user interface consequently supports search, filter by ontology, anatomy, and metadata, visual browsing of tissue samples, and right now we have the initial tools that Vanderbilt shared. Recently, Vanderbilt uploaded a new data set, which is not yet covered here. You also have a way to do visual browsing of these tissue samples across different levels. You can connect with data authors to inquire about technology details or about the data itself. You can download the data from different levels, and you can also submit questions and comments on the CCF user interface. The GitHub repository, this is all open free code. You can go to that GitHub repo and check out the code. We also have a 6-week-old development interface, so that's partially functional. After 9 weeks, it will have all the functionality we had in the initial specification, which you can see up here. And so again, if you're interested, please do check out these links and send us more questions and comments on it as well. Here you see some screenshots. So you have a whole body view, which can also be part male, part female. You have the organ view, you have the tissue view, which very much looks like a photo browser, and on purpose so, because many people know how to use those. And you can then zoom into these beautiful tissue slices that came from Vanderbilt, and that's very much like a Google map. However, right now there are 2 slices and you can zoom in and out. That's all we have right now. So we can't wait to actually have this rich data, which we just saw from that team. And Jeff had some of this data this morning. It's just not ready yet for us to have integrated here. The interface itself, as you can see, supports semantic search, uses the ontologies that you saw in Paul's presentation this morning to support semantic browsing. We also have semantic filters, for instance, on the different technologies that Vanderbilt is using. You also see that there are different types of imagery that you can select from, and we have a little bit of information on what they mean in case you're not a wet lab bench scientist. The interface that you just saw is very much inspired by other efforts, which look also at how to make data sets easily browsable and downloadable. Initially, we believe that people will come to HuBMAP and just download everything, but everything is actually already quite big. So I think we want to make it easier for them to look through the different types of data sets that soon will become available. In terms of year 2 plans, which actually starts June 21st, our year 2, we will develop, test, and optimize the different tissue registration user interfaces and optimize them for different HuBMAP organs. I believe we will need different ways to register for different organs. We will evaluate and enhance the functionality of CCF user interfaces. So we are actively looking for power users, which might be able to help us understand how HuBMAP data might be used in the future for research or clinical applications. We will, together with Jeff and Sanjay and others working on kidney data, serve a kidney microatlas and also run formal user studies on both types of user interfaces. In addition to this, we propose to research and develop a visual human massive open online course. In other work we are engaged in, we noticed that open data and open code goes extremely well with open education. Oftentimes you need a little bit of hand-holding to understand, for instance, the quality and coverage of data, the utility and proper usage of tools and workflows, and also to demonstrate new single-cell analysis and mapping techniques. In addition, we will also host another CCF workshop together with our HuLS team, either in fall or in spring 2020. I will conclude with a set of open questions. We are still not perfectly clear on what data sets and unified formats will become available when. We have started a longer Google Doc to just help TMCs understand what data formats are good on our side, and we would love to have those if possible. We also need to understand what tasks existing ontologies perform well and which ones are not so well covered. And ultimately, we are still reviewing other user interfaces to see what they perform well and where they do fall short. So I'm ready to open for questions, and thank you for your attention.
Any questions?
Who is a power user?
Yeah. So, for instance, Leonard recently visited Vanderbilt, and Jeff connected him with nephrologists. And I think it's very interesting to understand what nephrologists would like to do with the HuBMAP data. I believe if Leonard would talk with postdocs, younger postdocs, which want to mostly publish and get a tenure track position, I think he would get a different user story. There are also technology experts like Jeff himself who need to understand what the space of new technology developments looks like. So that's again a different power user. And right now we have not talked with clinical researchers which want to improve health, which is of course a major goal for NIH in general. So I think they would have again different use cases. And ultimately it might be that we have a standard interface that's easy to use for general audiences, but then have very specialized interfaces for the four user groups I just mentioned.
Hi, this is Todd. I just had a question about the image registration, which is becoming a very powerful approach for many people, and it's very interesting. I'm wondering if you could talk a little bit about how much time it takes and whether you anticipate that type of approach is going to be something that you can train lots of people to do to aid in the project.
So there are, of course, approaches like where you train citizen scientists to do some of this. But there's, for instance, Edge the cell, where anyone can get trained to indicate the outlines of cells, and you do this across the tissue stacks. And ultimately, you can then extract 3D structures this way. For what I shared here for the registration of tissues inside of an organ, we were planning to do this together with surgeons, which are actually involved in extracting the tissues currently. It's not totally clear yet how many times that needs to be done by a human being before machine learning can help substantially, maybe initially just as a guide, ultimately maybe 100% autonomously. So this all will also very much depend on human diversity we try to capture here. As you might have seen, just for the kidney, there's quite a bit of diversity in the just gross anatomy. I do not yet know how much diversity there will be at the subcellular level or the cell type level, and how these cell types ultimately cluster and are different across individuals. I think we're just learning about this as we go. So I'm not sure if this is the answer you wanted, but that's the answer I have.
It's all that we could say at this point. Thank you.
Thank you. All right.
Great.